Our today's topic for discussion, it is the lipid metabolism. We have studied the chemistry of lipids, the different types of lipids. Then we have also studied the phospholipids, the different types of phospholipids, sphingolipids, etc. Okay. Now, naturally, when lipids they are taken in the diet, after they have taken in the diet, they will get digested, they will get absorbed. And after absorption, these lipids, they must be utilized by the body. Naturally, the major function of the lipid, as we have seen, it is the release of energy. But for release of energy from the lipids, they have to be oxidized in the body. So after absorption, lipids, they are either oxidized in the liver or stored in the depot fat. So after absorption, what will happen to the lipid? They are either oxidized in the liver or stored in the depot fat. For lipids utilization, triglycerides, they are first hydrolyzed by lipases to glycerol and free fatty acid. Okay, so triglycerides, they will get acted upon by the lipases and they will get converted into what? Glycerol and free fatty acid. Glycerol, either it will get converted to hexoses or enter glycolysis after phosphorylation. So this glycerol, either it will get converted to hexoses or it will enter glycolysis after phosphorylation, and it will get metabolized to CO2, H2O with large amount of energy. Naturally, after phosphorylation, it will get metabolized to what? To CO2, H2O, and there will be release of what? Large amount of energy. So naturally, we should understand what will be the fate and utilization of the fat after absorption. So what are how the body, it utilizes the fat after absorption. One of the most important utilization, that fat, it is stored as the depot fat. Okay, and complete second utilization, complete oxidation to liberate energy, CO2 and water. So lipids, either they will get stored as a depot fat or there will be complete oxidation of these lipids to liberate energy, CO2 and H2. Now, as in the chemistry of lipid and functions of the lipid, we are knowing that the lipids, they are the structural elements of the cell membrane and cell component. Okay, so such as lecithin, cephalin, sphingomyelin, cerebrosides, etc. They are what? They are the structural elements of the cell membrane, phospholipids, and of the cell component. So this is also one of the utilization of a fat. Now, one more utilization of a fat, formation of a milk fat. So during lactation period, the mother's milk, it is the major source of food to the infant. So naturally, the formation of a milk fat from the fat that will get absorbed from the body, it is also one of the fate or one of the utilization. Now, the lipids, they will be also utilized for what? For synthesis of a cholesterol, for the synthesis of the steroids, and for the synthesis of the phospholipids. As we know, the cholesterol, it is the one of the essential, or you can say the essential substance to the body. So if this cholesterol, it is not present in the food, naturally body will synthesize the cholesterol. So that will be the endogenous synthesis of the cholesterol that will take place. There will be synthesis of the steroids from the fat. There will be synthesis of the phospholipids. And all these three, they are having there some important, you can say, functions, so many important functions in the human body. Now, one more utilization of a fat, they will get converted to carbohydrate, amino acids, etc. Now, from these fats only, there is a synthesis of a prostaglandin, there is a synthesis of the bile acids, and there is a synthesis of a bile salt. So, bile acids, they are the glycocholic acid, taurocholic acid, like that. The bile salt, sodium glycocholate, sodium taurocolate, potassium glycocholate, potassium taurocolate. Then synthesis of prostaglandins, they are the local, you say, hormones. So they are having their wide variety of actions. Bile salts, they will be necessary for what? For the digestion and absorption of a fat. Specifically, the emulsification of a fat that is very much required for the action of the lipolytic enzymes. And these bile salts, they will reduce the surface tension from the lipid molecules. They will make them sparingly soluble in the water, or there will be fragmentation of the lipids that will take place by the bile salt, or there is a formation of a micelles so that the lipolytic enzymes, they will act on that. Okay, so naturally all these are what? All these are the fate of a lipid or fate of a fat or utilization of a fat for absorption. Is it clear? Now coming to the what? The major pathway for the oxidation of a fatty acid. When we say that the fatty acids, they must be utilized in the body or they are oxidized in the body to liberate large amount of energy, the major pathway for the oxidation of a fatty acid, it is the beta oxidation. Okay, so the major pathway for the oxidation of a fatty acid, it is the beta oxidation. Now, what do you mean by beta oxidation? The oxidation that takes place at the beta position. What is the beta position? The third from the carboxyl group. So that position, which is third from the carboxyl group, it is the beta oxidation. And as the oxidation, it takes place at the beta position, it is named as what? Beta oxidation. Okay. Now, it is invented. Who is the scientist who has invented this? The Knoop, Knoop. Knoop is the scientist who has invented the beta oxidation, or he has invented this pathway. Okay. Now, oxidation of a fatty acid at beta position, it results in what? Results in elimination of a two-terminal carbon atom. So when beta oxidation takes place, with one term, there is an elimination of a what? Elimination of a two carbon atoms. Leaving behind what? Two terminal carbon atoms as acetyl-CoA and leaving behind the fatty acyl-CoA or fatty acid, which will have two carbon atoms less than the original fatty acid. I will again repeat that why it is called as beta oxidation, because the oxidation, it takes place at the beta position. Which is the beta position? Beta position, it is third from the carboxyl group. Who has invented this cycle? Knoop is the scientist who has invented this cycle. So when beta oxidation, it takes place, there is a removal or elimination or removal of a two terminal carbon atoms in the form of acetyl-CoA. So when one round of a beta oxidation, it will take place, there will be removal of the two terminal carbon atoms in the form of acetyl-CoA, leaving behind the active fatty acid or leaving behind the fatty acyl-CoA or fatty acid, which will have two carbon atoms less than the original one. Okay, this was the introduction to the beta oxidation. Now, the standard example that is taken for the understanding of the beta oxidation, it is the 16 carbon fatty acid, that is the palmitic acid. Okay, so always the standard example that is taken for the beta oxidation, it is the palmitic acid. So this palmitic acid, it is what? It is the 16 carbon fatty acid. Now this beta oxidation, it involves 5 steps or it involves 5 reactions. The first reaction, it is the activation of the fatty acid. Second, it is the dehydrogenation. And third, hydration. Hydration, you understand, addition of a water molecule. Dehydrogenation, removal of the hydrogen molecule. Fourth reaction or fourth step, again it is the dehydrogenation. And the fifth, it is the thiolytic cleavage. Is it clear? So there are five important steps of the beta oxidation. First is what? Activation of the fatty acid. Second, dehydrogenation. third hydration, fourth dehydrogenation, and fifth thiolytic cleavage. What is the standard example we are taking? That is the 16 carbon fatty acid or the palmitic acid. So the first step, it is the activation. So 16 carbon fatty acid plus OA plus ATP. Okay, so ATP will be required. Two ATP molecules that are required for the activation, and the enzyme that is required for this reaction to take place for the activation, it is the thiokinase. And when the activation it takes place, there is a formation of what? Fatty acyl CoA or active fatty acid plus AMP plus pyrophosphate. That is adenosine monophosphate and pyrophosphate. So the first step of the beta oxidation, it is the activation, which is the enzyme thiokinase. So 16 carbon fatty acid plus OA plus ATP, enzyme is thiokinase. There is a formation of a active fatty acid, okay, or fatty acyl CoA. But naturally, what happens? So first step it is the activation, but the enzymes of this beta oxidation, the other enzymes of the beta oxidation, they are present inside the mitochondria. Okay, and this activation it takes place outside the mitochondria. Now, this activated fatty acid, it is impermeable to the mitochondrial membrane. Means it cannot enter inside the mitochondria. And where the other enzymes they are present inside the mitochondria. So to transport this activated fatty acid inside the mitochondria, there is a need of a transporter. And this transporter, it is the carnitine. Okay, so carnitine, it is the transporter that will be required for what? For the translocation or for the transport of activated fatty acid inside the mitochondria. And why it is needed? Because the enzymes of the beta oxidation, they are present inside the mitochondria. So always when beta oxidation it is to be understood, we have to remember the role of carnitine. So carnitine, it is the transporter. It will join with the activated fatty acid, and this activated fatty acid, it will come in the inner side, or it will come inside the mitochondria. Is it clear? Why it is needed? Because the other enzymes of the beta oxidation, they are present inside the mitochondria. So the first step we have understood, that is the activation, then translocation or transport of the activated fatty acid inside the mitochondria. So carnitine, it plays one of the important role for the translocation or transport of the activated fatty acid inside the mitochondria. So inside the mitochondria, now there is a activated fatty acid or fatty acyl CoA. Now what is the second step of this beta oxidation? It is the dehydrogenation. Okay, so the second step of the beta oxidation, when the activated fatty acid it has come inside the mitochondria, it is the dehydrogenation. And the enzyme that is required for this step, it is the acyl dehydrogenase plus FAD. And there is a formation of a what? Alpha beta unsaturated activated fatty acid. So there is a conversion of FAD to FADH2. It is the dehydrogenation, and there is a formation of a what? Alpha beta unsaturated activated fatty acid from the fatty acyl CoA. First step activation, second step dehydrogenation. So there is a formation of a what? Alpha beta unsaturated activated fatty acid, FAD2 get converted to FADH2. Is it clear? So it was the second step of beta oxidation. Now the third step of the beta oxidation, it is the hydration. Hydration, you understand. Hydration means what? Addition of a water molecule. And the enzyme that will be responsible for this step, it is the hydratase. Okay. So the third step, there is the hydration, it will take place. The enzyme is hydratase plus H2O. and there is a formation of a what? Beta hydroxy activated fatty acid. Is it clear? Or beta hydroxy alkyl CoA. So the third step or the third product that is formed in the beta oxidation, it is what? It is beta hydroxy activated fatty acid. Okay, is it clear? Now, the fourth step, again, it is the dehydrogenation. First step activation, second step, then you can say dehydrogenation, third step hydration, and fourth step, again, it is the dehydrogenation. And the enzyme that will be responsible for this step, it is beta hydroxy alkyl dehydrogenase. I will again repeat, the enzyme that is required for the dehydrogenation, it is what? It is beta hydroxy alkyl dehydrogenase plus NAD. And there is a formation of a what? Beta keto activated fatty acid. So first it was the activation, and you can see in the fourth step there is a formation of a what? Beta keto activated fatty acid. Now the fifth step and the last step of the beta oxidation, it is the thiolytic cleavage. What is the fifth step? It is the thiolytic cleavage. And what is the enzyme that is responsible for this reaction to take place? It is thiolase. So there is a formation of a what? The 14-chain activated fatty acid plus acetyl-CoA plus thiolase plus CoA. Means the fifth step, it is what? It is the cleavage. There is a formation of a what? 14-chain activated fatty acid plus acetyl-CoA. Now this acetyl-CoA will undergo, it will enter into the what? Into the TCA cycle. And in TCA cycle, it will get completely oxidized to what? To water, to H2O, and the energy release. Okay. Now, so naturally, by the beta oxidation, it takes place inside the mitochondria, because it takes place in the mitochondria of the liver, because the enzymes of beta oxidation, they are present close to the enzymes of what? Enzymes of the electron transport chain. Why there is a need of, you can say, activated fatty acid to get back into the what? Inside the, you can say, in the cells, because there is a what? Because there is a need of what? The transport mechanism, the carnitine. So the beta oxidation, it will take place in what? In the mitochondria of the liver, because the enzymes of beta oxidation, they are present close to the enzymes of what? Enzymes of the electron transport chain. Okay, so the oxidation, it will take place in what? Inside the mitochondria of the liver, because the enzymes of fatty acid oxidation, they are present close to the enzymes of what? Enzymes of the electron transport chain. Okay, now coming to the what? Coming to the energetics. Coming to the what? How much you can say, how much amount of energy that will get liberated when beta oxidation it will take place. Okay, so as we know that it takes place in what? The beta oxidation, it takes place in the mitochondria of the liver, because the enzymes of the beta oxidation, they are present close to the enzymes of what? Enzymes of the electron transport chain. Okay. Now coming to the what? Coming to the energetics. Energetics, the word you understand, how much energy that will get liberated when there will be a what? Complete oxidation of a what? Of one molecule of the, you can say, of the palmitic acid. Okay. So oxidation, as it will take place at the beta position, third from the carboxylic group, it will be the beta oxidation. Now coming to the what? Coming to the how much ATP that will get released, or how much ATPs that will get formed with one turn of beta oxidation. And what is the standard example? I have told it to you. It is the palmitic acid, which is 16 carbon fatty acid. Okay. So first palmitic acid, which is the common, you can say, lipid that is present in the food or in the diet. So palmitic acid, it will undergoes, you can say 7 times, or bit of it will underwent with the bit oxidation, and there is a formation of how many acetyl-CoA molecules? 8. Okay, so 8 ATP molecules they will be formed. Now each acetyl-CoA will go inside the, you can say, TCA to give what? To give the 12, you can say 12 molecules of ATP in one turn. Means with one turn of the beta oxidation, there is a formation of a what? There is a formation of the one molecule of acetyl-CoA. When 16 carbon fatty acid we have taken as a standard example, there is a formation of 8 acetyl-CoA molecules. So these 8 acetyl-CoA molecules, they will enter inside the TCA cycle or tricarboxylic acid cycle, and there will be liberation of how many ATPs there will be there? 12. So how many rounds they will be required for what? For the complete oxidation of a palmitic acid? So 7 rounds they will be required. In one round, the terminal 2 carbon atoms they will be removed in the form of what? In the form of acetyl-CoA. So there will be a what? There will be, so 7 rounds they will be needed for complete oxidation of a what? One molecule of a palmitic acid. Is it clear? So in one turn, 2 molecules of ATP they will be produced. So for a, you can say the 16 carbon fatty acid, 8 rounds of beta oxidation they will be required. Okay. So in one turn of coming to the major energetics, in one turn of a beta oxidation, with, you can say, there is a formation of a what? In one turn of a beta oxidation, there is a formation of how many ATP molecules? There is a formation of a 5 ATP molecules. And how many rounds of beta oxidation they will be required for complete oxidation of a 16 carbon fatty acid? 7 rounds they will be required. So 7 into 5, 35. Okay. So 7 into how much? So 35 ATP molecules they will be produced. Now each acetyl-CoA, it will undergo what? It will undergo TCA cycle. And with one turn of a TCA cycle, there is a formation. How many ATP molecules? 12 ATP molecules. How many rounds they will be required? 8 rounds they will be required. So 12 into 8, 96. And what is the, you can say, the, so there is a formation of what? The 7 into 5, 35, and these 12, with one turn of a TCA, there is a formation of how many ATPs? 12. So there will be formation of what? How many? 96. 12 into 8, 96 plus 35. There is a formation of what? The formation of the 131 ATP. How? 12 into 5, 12 into 8, 96, 7 into 5, 35, 35 plus 96, there is a formation of how much ATPs it will be there. So there will be formation of a 96. So 96 plus 35, there is a formation of how much? 131 ATP molecules. Out of these 131 ATP molecules, the 2 ATP molecules, they are utilized in the first step of activation. So net gain of energy in the form of ATP that will be obtained from what? From the one molecule of you can say the lipid. It is how much? It is 100 and you can say 129, because 2 molecules they have been utilized in the first step. So when there is a complete oxidation of a fatty acid, it will take place. There is a formation of a what? Of a palmitic acid. there is a formation of you can say lots of amount of energy. Is it clear? So this was about what? The beta oxidation. When we talk about the beta oxidation, naturally the standard example we have taken, it is the palmitic acid, which is the even number of carbon atoms that are there. Okay. So palmitic acid, it is taken as a standard example. The rest of the we can change the. Okay. Now, naturally, when we talk about this beta oxidation, we have taken oxidation of a fatty acid with even number of carbon atoms. But you can say there can be, or you can say the examiner may say that do the work for what? For the, not only that the, you can say, but there will be a, okay. Now, what happens to the what? To the oxidation of a fatty acid with odd number of carbon atoms. So fatty acid oxidation with what? With the number of, you can say, with add number of the, you can say, the carbon atoms. So fatty acid, 16 carbon fatty acid, it will undergo beta oxidation. There is a formation of acetyl-CoA plus propynyl-CoA. Propynyl-CoA will get converted to methyl-malonyl-CoA, and methyl-malonyl-CoA will get converted to succinyl-CoA, which can enter into the, you can say, into the PC cycle, or which can enter to form what? To form the protein. Okay. So Krebs cycle, fatty acid synthesis, the ketone body formation, etc. These are all the utilization of a fat before it will get deposited into the body. So fatty oxidation, there is a, you can say, beta oxidation, which is the commonest thing. There will be formation of acetyl-CoA plus propynyl-CoA. This propynyl-CoA, it will undergo, you can say, it will get converted to succinyl-CoA. This succinyl-CoA, it is also one of the important one. It will help in what? In understanding the other. So propynyl-CoA plus methyl-malonyl-CoA plus, you can say, succinyl-CoA, it will go to the TC cycle rest again. Then you can say the fatty acid synthesis not required. Okay. Then there is a, you can say, operative which cycle? The fatty acid synthesis, the cholesterol synthesis is required, and the, you can say the, okay. So it was about what? About the beta oxidation. And what we have taken the standard example, we have taken the standard example as what? As the palmitic acid. Is it clear? If at all there is any doubt, we can proceed further and we can what? We can see with the other doubts. Time कितना हुआ? Okay. Now, suppose there is a, you can say, what is the metabolic rate of acetyl-CoA? Because one of the major products that will be formed during the beta oxidation, it will be the acetyl-CoA. So what are the sources of this acetyl-CoA? Glucose, it will get converted to pyruvate, pyruvate to acetyl-CoA. Fatty acid, it will get converted to what? To acetyl-CoA. So then how this acetyl-CoA it will be utilized? It will be sent to the Krebs cycle. What do you mean by Krebs cycle? Breakdown of the fat into, you can say, okay. So there will be a, you can say, so when there is a, you can say, the ATP production, for that we have seen that the fatty acid or activated fatty acid, it will get converted to what? To the without number of carbon atom. So there is a format glucose to pyruvate, pyruvate and fatty acid, they will get converted to the acetyl-CoA. And this acetyl-CoA will enter the TC cycle to give what? The difference. Then it will be utilized for a fatty acid synthesis, for the cholesterol synthesis, and for the ketone body synthesis. Okay. This is the utilization of a, you can say, acetyl-CoA after the, you can say. Oh, is it clear? So fatty acid is beta oxidation. There is the formation of acetyl-CoA plus propynyl-CoA. Propynyl-CoA to Methylmalonyl-CoA, methylmalonyl-CoA to succinyl-CoA, and succinyl-CoA will enter into the what? Into the TCA cycle. So from the examination point of view and from health point of view, try to remember the different aspects of what? Of this acetyl-CoA. Okay. So this was about the today's topic. What do you mean by what will happen to the, you can say, to the food item that will be. So the digestion and absorption of the, you can say, broadly with the lipids. Then I have told you about the utilization, then how the, you can say. So this was about the detoxidation or the lipid, you can say, the oxidation of the lipids after digestion and absorption of the lipids. Thank you very much.
थोड़ा इधर घुमाने ये रिकॉर्डिंग वाला है ना क्या आ रहा है उस पे साइन
रिकॉर्डिंग